In this lecture, we're going to continue talking about cell transport, but this time I'm going to talk about osmosis. And this is not how you study for test in this class. It's an actual scientific thing, but it does not help you pass this class. So no trying to learn by osmosis. Osmosis is really simple to talk about, but it gets complicated because I can talk about it in a couple of different ways. Simply, osmosis is the diffusion of water through a semi-permeable membrane. It works just like diffusion does. However, I'm talking about one very, very specific thing. The only thing I care about when I talk about osmosis is water. I care about which way my water is moving. So it's the diffusion of water through a semi-permeable membrane like a cell membrane or the nuclear membrane or any kind of membrane you can think of. Just so I make sure that you know exactly what I'm talking about, I am going to give it a much more technical definition to it. Water flows down its concentration gradient until it reaches equilibrium. Just like my sugar particles I talked about in the diffusion lecture, my sugar will move out and it will flow down its concentration gradient. It will go from an area of high concentration to an area of low concentration until it reaches equilibrium, which means that I have an equal concentration of sugar throughout. For osmosis, I only care about water. So water will flow down its concentration gradient. It will flow from an area of high concentration to an area of low concentration until the water concentration on both sides of the membrane are equal, are at equilibrium. Now this may seem really easy right now, but it gets hard when I start talking about the different types of solutions that you can have in terms of this. The easiest one to talk about, and we'll start with this one, is isotonic. What does it mean if I have an isotonic solution? Well, hopefully you know or might recognize iso. Iso means same. So an isotonic solution is when the two solutions are the same concentration on both sides of the membrane. If I drew a picture of this, say I have what we call a U-tube, and in the middle of this U-tube, I have a semi-permeable membrane. This membrane only, in the case of osmosis, only lets water through. It's the only thing that can pass. I'm not talking about any particle or any molecule besides water. Water is the only thing that can come and go through that semi-permeable membrane. So say I have two different solutions in my U-tube. On this side, I have a solution that is 10% salt, therefore I'm 90% water. If I wanted to have an isotonic solution, I'd have to have the exact same thing on the other side. So this side of my U-tube would also have to be 10% and 90%. So that I would have an isotonic solution, the same solution on both sides. Now, does that mean that my water isn't moving through my semi-permeable membrane? No. I always have molecules moving, but my molecules are at equilibrium. So they're just, for every one molecule that goes to the left, I have one molecule of water that goes to the right. But this would be an isotonic solution. Your other option that you can have is a hypertonic solution. If I am hypertonic, it means the concentration of my solute, and these are my particles, my salt, my sugar, whatever is in there, is higher than the concentration of my water. It is hyper. I have more. On a U-tube, it would look something like this. Still got my semi-permeable membrane, and I'm going to have two different solutions in my U-tube. One over here, one over here. A hypertonic solution means that I have more salt, more sugar than my water. So say on this side I had 60% NaCl and I have 40% H2O. I have a hypertonic solution. This is considered to be the hyper. On the flip side of this, I can have also the opposite of hyper is a hypotonic solution. This is when the solute concentration, my particles, is lower than the water concentration. So if I go back to my YouTube, say on this side, I have 40% NaCl and I have 60% H2O. This is going to be considered my hypo solution. When I have this type of solution, remember the only thing I care about is which way my water moves. That's the only thing that can pass through the semipermeable membrane. And it always flows down your concentration gradient. So if you took a look at this, I have 60% H2O on one side, 40% H2O on the other side. Which way will my water move? It will go down my concentration gradient. I have to go from 60 to 40%. So eventually, water level on this side will increase. My water level on this side will decrease. Now, why does this matter? Why am I making you know this? It all has to do with osmotic pressure, which is really important to cells. I'm going to talk about this in class, but I want you to try to figure out what type of environment I'm about to put my red blood cells in terms of tonicity. Is it isotonic, hypertonic, or isotonic? Osmotic pressure has to do with red blood cells. So RBCs, red blood cells. If I drew a red blood cell over here, my red blood cell has a specific concentration roughly. 99.1% of my red blood cell is going to be made up of water. The other 0.9% is made up of salt, NaCl. If I take my red blood cell and I put it into a beaker full of a solution, I want you to tell me in terms of tonicity, isotonic, hypertonic, or hypotonic, what is going on with my cell. Say my solution over here is 90% H2O and 10% NaCl. Which way is my water going to flow? Because I'm still talking about osmosis. So on this drawing, I want you to tell me which way am I going to go, and then tell me if I am hypertonic, isotonic, or hypotonic. I want you to tell me one of those things. Show me which way my water is flowing, and tell me what tonic I am. Do it for this one as well. I have another beaker that's got another solution in it, and I place my red blood cell into it. It's just this time, my solution is 100% H2O. Tell me what's going on in my cell. Am I hyper, hypo, or isotonic? And tell me which way my water is flowing. And then we will talk a little bit more about this in class when I see you.